Hello guys, this is Vineet and welcome to Molecular Biology series. Today I'll be discussing about the histone variants and their role in epigenetics. Okay, so before we get into the actual topic, let us try to understand the basics of this epigenetics. Okay, so epigenetic is commonly referred to as molecular pathways, all molecular pathways modulating the expression of a genotype into particular phenotype. Okay, so what we are doing is we are modifying the expression of a gene, okay, and passing this modified expression from one generation to another generation. Okay, so this kind of transferring of modified expression of a gene, okay, is commonly referred to as the epigenetics or epigenetic inheritance. Okay, and the epigenetic inheritance mainly results from the inheritance of a chromatin state. Okay, whether the chromatin is in euchromatin state or the heterochromatin state. Okay, so this kind of transferring the modified expression from one generation to another generation is commonly referred to as a epigenetics. Okay, and information that is passed from one generation to another or the next generation or the daughter cells, okay, so during the cell division, this is not encoded in the DNA sequences. Okay, that means we are not doing any kind of mutations like insertion, deletions, or the transcription things. Okay, we are just modifying the expressions. Okay, that means the suppression of the gene transcription or the repression of the gene transcription will be done in this epigenetics. Okay, so this kind of modification will be done, and a lot of molecular pathways involved like chromatin remodeling, covalent modifications of DNAs, and covalent modification of the histone proteins. Okay, and histone chaperone depositing. Okay, the whatever the histone chaperones are there, they deposit typical variants of the histones or the canonical histones onto specific sites. Okay, so they regulate the gene transcription or the expression of that gene or the repression of that gene. Okay, so these are called as the this is the basic information about the epigenetics. Okay, so if we talk about the major molecular pathway which is involved in the epigenetics, that is the covalent modification by different types of enzymes. Okay, if you take this DNA, there will be some kind of DNA methyl transferases. They transfer a methyl group on the cytosine nucleotide, or they can also do this hydroxy methylation. Okay, and this kind of methylation can be seen only in the CpG islands. Okay, CpG islands, they are cytosine per guanine rich sequences. Okay, they are just specific sequences only. They contain huge amount of cytosine per guanines. Okay, so these are called as a CpG islands. Okay, and most of the methylations will be seen on the cytosine nucleotide. Okay, and if we talk about the histone modifications, okay, so we also have a lot of methyl transferases and acetyl transferases, also the phosphorylating proteins. Okay, so most of the histone proteins, they undergo this kind of modifications on the lysine and arginine amino acids. Okay, and also the serine amino acids. Okay, these kind of covalent modifications which are happening on the different type of amino acids or nucleotides. Okay, so they play central roles in most epigenetic inheritances. Okay, and we also have the second major molecular pathway that is called the chromatin remodeling. Okay, the basic nucleosome sliding model by this switch and snip proteins. Okay, which are the second family of this chromatin remodeling family members. Okay, so the switch and snip proteins, so they do this chromatin remodeling majorly, and also the histone chaperones. Okay, HWA family, ISWA family, and a lot of histone chaperones. Okay, so these histone chaperones, they replace a histone variant with the canonical histone, or they place the canonical histones at specific sites so that the heterochromatin will be stayed in the heterochromatin form only, or the euchromatin will be converted into heterochromatin. Okay, so these kind of changes are referred to as the chromatin remodeling. Okay, so the chromatin remodeling also get transferred from one generation to other generation, but not always. Okay, so chromatin remodeling is not always inherited, and not all epigenetic inheritance involves the chromatin remodeling. Okay, so you have to remember this point. Okay, and so when doing the cellular reprogramming, that means whenever we try to make this pluripotent stem cells in the laboratory. Okay, so whatever the epigenetic marks are there, okay, whatever the covalent modifications are there on the DNA or on the histone proteins, okay, so they are all removed. They are all removed from the differentiated cells. Okay, and these differentiated cells further replaced with the pluripotent stem cells. Okay, so this is what we do in the cellular reprogramming. Okay, in the laboratories. Okay, yeah, if we talk about this DNA methyl transferases, okay, which methylate the cytosine sequences in the CpG sites or the CpG islands. Okay, they add a specific methyl group onto this cytosine nucleotide. Okay, so and convert this normal cytosine into 5-methyl cytosines. Okay, there are a lot of enzymes, DNA methyl transferase 3A1, 3A2, and 3B. Okay, so these are the major enzymes which are found in the nuclear genome. Okay, and these enzymes majorly works on more than 3000 regions, 3000 different regions in the nuclear genome. Okay, so there are 3000 specific regions for this different methyl transferases in the nuclear genome. Okay, where they do this methyl group transferring activities. Okay, so these are what the some basic informations about the epigenetics. Okay, and the major molecular pathways involving will be the covalent modifications and the chromatin remodeling. Okay, chromatin remodeling can be done by nucleosome sliding, or the recruitment of histone chaperones for depositing canonical histones or the histone variants. Okay, a lot of things happens in the chromatin remodeling. Okay, and one more major covalent modification is the DNA methylations, basically, which happens on the cytosines. Okay, so this is what about the epigenetics. Okay, and if we talk about the normal histones, okay, so these normal histones, they are synthesized only during the S phase. Okay, that is the DNA replication phase. Okay, and are deposited onto the newly replicated strands. Okay, so whenever the DNA replication is happening in the S phase, okay, so the nucleosomes which are present on the parent strand, okay, so they are broken down. They get broken apart into different types of histones. Okay, histone tetramers. Okay, this nucleosome core actually breaks into two different tetramers. Okay, one is H3 and H4 tetramer, and one is H2A and H2B dimers. Okay, two dimers will be there. Okay, so this kind of breaking, or this kind of disruption, okay, will provide the histone proteins for the newly synthesized DNA strand. Okay, so these histones, they further get recruited by this CAF1 histone chaperone. Okay, so this is a chromatin assembly factor 1. This histone chaperone takes the disrupted histones from the parent strand and places them on the newly synthesizing strand. Okay, this kind of placing also involves a lot of different factors known as the histone binding proteins, DPB3 and DPB4 complex, okay, which are associated with the polymerase enzyme, DNA polymerase epsilon. Okay, so all these enzymes, okay, all these protein molecules all together, they deposit the histones on the newly synthesized strands. Okay, so since the DNA replication will form two new daughter DNA strands. Okay, so one daughter DNA strand will get the parent histone molecules, or whatever the broken histone proteins are there, they will be recruited on the parent one newly synthesized double stranded DNA strand. Okay, but the other double stranded newly synthesizing double stranded DNA strand, okay, will involve de novo histone depositions. Okay, that means new histone molecules will be synthesized during this replication phase. Okay, and these newly synthesized histone molecules will be recruited onto the second newly synthesized DNA strand. Okay, so this kind of a fresh recruitment is what we call that as a de novo recruitment. Okay, or the de novo placement of this histone proteins. Okay, so this is what about the partitioning of histone proteins during the replication work, or during the replication process. Okay, and the whatever the mRNAs that are coming out from the genes, okay, the gene clusters of these histone proteins. Okay, so these mRNAs are majorly stabilized by this U7 small nuclear riboprotein complex, okay, ribonuclear protein complex, and also the stem loop binding proteins. Okay, this stem loop binding proteins, and as well as this U7 small nuclear ribonuclear protein complex, okay, they stabilize the mRNA molecules. Okay, and they promote this histone synthesis. Okay, multiple histone synthesis during the DNA replications. Okay, this is what about the special thing about the histone mRNAs. Okay, this kind of stabilization you won't be seeing any kind of other mRNAs. Okay, most of the mRNAs contain the 7-methyl guanidine cap and the 3' polyadenyl A tails. Okay, polyadenyl tails. Okay, but these histone mRNAs, they are stabilized by this these two different proteins. Okay, the one is U7 small nuclear ribonuclear protein complex. Okay, this complex stabilizes the mRNA, and also the stem loop binding proteins. Okay, so they help tight coordination of histone synthesis. Okay, the multiple histone protein synthesis will be happening during the DNA replications. Okay, so this is what about the normal histones, okay, which we call that as a canonical histones. Okay, and histone chaperones, they also direct the epigenetic information. Okay, that means they transfer the newly formed chromatin state, okay, from the parent cell to newly synthesized cell, okay, the daughter cell. Okay, so this kind of transferring, that we call that as epigenetic information. Okay, and most of the histone chaperones, they do this remodeling things. Okay, so one major remodeling chaperone, histone chaperone, is what CAF1. Okay, so this CAF1 has one major large subunit in it. Okay, so this subunit the larger subunit of this CAF1, okay, it dimerizes and bind to PCNA, okay. So the PCNA is a unwinding ring which will be seeing in this, the which we'll be seeing in the helicase actions, okay. This PCNA and dimerized to CAF1 will now bind and allow the formation of this H3, H4, ASF1 and CAF1 complex, okay. So this complex contains two chaperones as well as two histone molecules, okay. This ASF1 is an anti-silencing factor 1 chaperone and CAF1 is a chromatin assembly factor 1 chaperone, okay. So this kind of 2:2 complex is formed due to this large subunit, okay. So whenever the large subunit get dimerizes in the CAF1, okay, it binds to the PCNA and allows the formation of this 2:2 complex, okay, that is H3, H4, ASF1 and CAF1 complex, okay. And this CAF1 complex promotes the deposition of the histones, that is H3 and H4, onto the newly synthesized DNA strands, okay. And we also have the different kinases, okay, which regulates the CAF1 histone chaperone, okay. So the regulation of this histone, the CAF1 chaperone, or the dimerization of this large subunits of the CAF1 chaperone is regulated by DBF4 dependent kinases, okay. So now one kind of a cell cycle kinase protein, okay. And we also have upside regarding this histone binding proteins, okay, DPB3 and DPB4, okay. So these proteins which are often regarded as the histone binding proteins, okay. So these are the subunits of this DNA polymerase epsilon, okay. That means they get associated with the DNA polymerase epsilon or epsilon protein, okay. And it also associated with the histone deacetylases and also the chromatin remodelers, okay, and plays a crucial role in the inheritance of this histone hypoacetylation in chromatin, okay. That means the silenced form of the chromatin material, okay, will be inherited from the parent cell to daughter cells, okay. So these proteins will be playing a major role in this kind of inheritance patterns, okay. The major inheritance is the silence to heterochromatin state will be inherited from the parent to daughters, okay. That is mainly due to this presence of DPB3 and DPB4 proteins, okay, which are often regarded as the histone binding proteins, okay. So this is what about the synthesis of this canonical histones and their deposition by CAF1 and ASF1 histone chaperones, okay, and their inheritance by this DPB3 or DPB4 histone binding proteins, okay. And if we talk about the histone variants, okay, so this canonical histones, they generally differ from this histone variants, okay. The synthesis of these canonical histones will be only seen in the S phase, okay, the synthesis phase of the cell cycle or during the DNA replication, okay. But these variant forms of these canonical histones, or what we call this as the histone variants, they are expressed throughout the cell cycle, okay. That means whenever a gene wants to get activated, or whenever it want to undergo transcription process, okay, at that time also this histone variants get synthesized inside the cells, okay. So that means irrespective of the replication phase or irrespective of the synthesis phase of cell cycle, okay, histone variants will be expressed throughout the cell cycle, okay. And some histone variants, they are tissue specific, okay. That means they only get expressed in the some parts of the brain or some parts of the testes, okay, but not in any other somatic cells inside the body. That is what the specificity goes for this histone variants, okay. And most of the histone variants even undergo post-translation modification, that is the covalent modifications like methylations or acetylations, phosphorylations or ubiquitinations, okay. So this kind of covalent modifications will also be done on this histone variants, okay. So which in turn regulate the chromatin remodeling or the transferring of the epigenetic information from parent to daughters, okay. So these kind of histone variants are majorly first discovered by this Franklin and Zweidler in 1997, okay. So they separated this histone variant from mammalian tissues by acid urea titron, okay. So doing this acid urea titron polychromate gel electrophoresis, they separated the canonical histones as well as the some different kinds of proteins, which are regarded as the histone variants, okay. And these histone variants, they are dynamic, okay. That means they always change their positions, okay. They always get exchanged with the normal histones, okay. So that their action is not particular or the consistent, okay. They are dynamic, okay. They change from one place to another, and period to period, okay. So that is what about the histone variants. Okay, and if we talk about the histone variants, okay, we have four major nucleosome core canonical histones, okay, which are present in the nucleosome core. Okay, and we also have another canonical histone, that is the H1 histone, which we often call as a linker histone. Okay, so these canonical histones are normal histones or the original histones. Okay, and they have the variants. Okay, and most of the variants are, you know, in or yeah, most of the variants uh, will be seen for this H2A variant. Okay, so H2A histone. Okay, so H2A contains most of the variants, and then the H3. Okay, so H3 contains around 7 variants. Okay, H3.3, 3, Y1, Y2, 3.4, 3.5, and CNPA, and H4 contains only one variant, that is the least histone variant containing canonical histone. Okay, so most of the histones, they only differ by single amino acid, or certain kind of extended polypeptide chain. Okay, so most of the variants, they differ only by one or two amino acids. Okay, and some histone proteins like macro H2A2, they differ from the original histones by containing extra polypeptide chain. Okay, extra polypeptide chain of around 200 amino acids. Okay, so this kind of macro H2 A1.2 or A2 or A1.1, they contain extra length of amino acid sequences. Okay, so that, and most of the H3.1, H3.3, and H3.1, this kind of histone variants, they only differ by one amino acid. Okay, that is serine and alanine amino acid, which will be presenting in this different histone variants. Okay, and H4 histone, most of the studies has not done on this histone 4G yet. Okay, so we know this histone 4G is present, but most of the functions are not known. Okay, and if we look at the structure of the histone molecules, okay, most of the canonical histones like histone 3, 4, and H2A and H2B, the common thing on this of these four histones is the presence of the helical fold domain. Okay, or which we also call the histone fold domain, which contains three alpha helices, alpha 1, alpha 2, alpha 3. So these alpha 1, alpha 2, alpha 3 helices, which are present in the histone fold domain, is common for histone 3, histone 4, histone H2A, and histone H2B. Okay, and most of the variants may contain the original histone fold domain, or they may undergo some kind of slight variations in the histone fold domains. Okay, but most of the canonical histones, they contain the similar kind of histone fold domains. Okay, no variation will be seen. Okay, and they also contain three alpha helices in the histone fold domain. Okay, and if you look at this histone 3, the variant form of the histone 3, H3.3, and centromere H3 variant. Okay, so these these two variants. Okay, there are also other variants, but here we have only discussed the structures of these two variants. Okay, these two variants, okay, they only differ by some kind of amino acids in the histone fold domain. Okay, and centromere has three histone protein. It contains two amino acid less polypeptide chain. Okay, that is, it contains only 133 amino acid polypeptide chain, while the H3.3 contains 135 polypeptide chain. Okay, and both H3 canonical H3 histone and H3.3 histone variant look same, but has some kind of variations in the histone fold domain as well as the N-terminal polypeptide chain. Okay, and the same goes with the H2A, H2AZ, H2AX, and H2B. They all contain similar amounts of the amino acids. But if you look at this macro H2A, it contains an extra polypeptide chain, that is about 200 or 200 to 220 amino acid extra sequence. Okay, this macro H2A histone protein, it contains an extra 200 to 220 amino acid chain. Okay, so that makes a long protein molecule. Okay, so because of presence of this macro H2A, the euchromatin will be converted into heterochromatin. Okay, so it further condenses the nucleosome core, so that the chromatin will be stayed in the heterochromatin form only. Okay, and most of the macro H2A will be present, will be seen in the gene inactive regions. Okay, wherever the heterochromatin regions are present, at that places the macro H2A will be seen. Okay, and we also have H2X. Okay, remember this X refers to the X chromosome. Okay, and Y, we have Y over here. So it refers to the Y chromosomes. Okay, that means H3.Y will be mainly seen in the testes, okay, that is in the Y chromosomes, okay, and H2AX, okay, it is mainly seen in the X chromosomes or the sex chromosomes, okay. And macro H2A, it is mainly present in the inactive regions, while H3.3 variant, histone variant, it is mainly seen in the telomere regions, okay. And centromere protein A, the name itself tells that it is present in the centromere region, okay. And along with this centromere protein A, histone 3.3 forms a heterotypic protein or heterotypic dimer. Okay, so these kind of heterotypic dimer will be seen in the pericentromere regions and the centromere regions, okay. Both regions contain this kind of heterotypic dimers, okay. That means H3.3 variant and another variant of this H3 variant, H3 histone, that is the CNPA, okay. So these both they combine and form a heterotypic dimers, okay. And in the gene active regions, that is in the case of euchromatin regions, okay, we'll be seeing this H3T in testes, okay, and H3.5 in the also in the testes, okay. And we have H3.5, H2AZ and H2AB, okay. So this normal versions, okay, the normal versions of the H2A, the normal variants of this H2A, histone 2A, okay. So they contain 2 or 3 amino acids less than the normal H2A proteins, okay. This H2AZ and H2AX, H2AZ actually contains 2 amino acids, while H2AX contains another 12 to 13 amino acids more, okay. So extra amino acids, okay. And in the DNA damage, the histone 3.3 will be playing a major role in the DNA damage, okay, recruiting a lot of DNA damage proteins or the DNA polymerases or the ligases, okay. So it normalizes the nucleosome core so that the polymerases and the ligases can bind on the DNA sequences wherever the DNA damage has occurred, okay. And then the telomere, I already said you this 3.3 will be seen, okay. 3.3 is also seen in the case of this active gene regions, okay. This is what about the localization of the histone variants and the canonical histones, okay, and the structure of this canonical histones as well as the histone variants, okay. And remember, histone variant linker histone, okay, it doesn't contain any kind of histone fold domain, but it contains a WHD, that is also called as a winged helix domain, okay. And this winged helix domain is characterized by the presence of alpha helices as well as the beta sheets, okay. So 3 alpha helices will be there and 2 beta sheets will be there, okay. And the length of the histone 1 protein is about 179 amino acids to 223 amino acids depending upon its variant, okay. So this is about the major basics of this histone variants, okay, and also the canonical histones, okay. And if you look at these histone chaperones which are involved in the deposition of these canonical histones, okay, as well as the histone variants, okay, most of the histone variants are specified by the specific histone chaperones, okay. That means the CAF1 histone chaperone, okay, it is involved in deposition of H3.1 and H3.2, while H3.3 involves Hira chaperone, ubiquinol chaperone and cabin chaperone, okay. So these 3 chaperones are required by this H3.2 to get deposited on the gene activating regions, okay. While the same histone variants of the H3, okay, H3.1 and H3.2 only requires chaperone, okay. That is the specific chaperones deposit the specific histone variants, okay. And H3, Y1 and Y2 on the Y chromosomes, they also get deposited by this Hira, UBN and cabin, okay. And while NAP2, which we also call as nucleosome assembly protein, this chaperone, okay, combining with another unknown chaperone, okay. So this is also test specific, okay. It deposits the H3.4 and H3.5 histone proteins onto the chromosome, okay, onto the gene active regions, okay. So this is what about the H3 histones, okay. And if we look at this H2A variants, okay, H2A variants mainly get deposited by the FACT, which you also call it the facilitates chromatin transcription, okay. This is also chaperone, FACT is also chaperone, okay. In the presence of this NAP1, okay, it deposits the H2A variant, okay, and FACT alone it deposits the H2AX, okay. And the deposition of this macro H2A is also done by this FACT, is also done by eviction of this macro H2A is also done by this FACT, okay. That means whenever the FACT goes into this heterochromatin regions, okay, wherever the compacted chromatin is there, okay. So whenever the FACT and ATRX proteins are recruited, okay, it evicts the macro H2. Okay, the eviction of the macro H2A is done by the FACT and ATRX, while the deposition of this H2A and H2AX variant, okay, the rest two variants of this H2 histone, okay, it is done by the FACT protein, okay, and the deposition of this H2AZ is done by another protein, another histone chaperone, that is P400 and SR cap, okay. So these P400 chaperone and SR cap chaperones, they deposit the H2AZ, while the eviction of this H2AZ histone variant will be done by ANP32E and as well as the INO80. Okay, so these two chaperones, they remove the H2AZ from the gene activating regions. Okay, while P400 and SR cap, they deposit the H2AZ variant on the gene activating regions. Okay, so the eviction and also the deposition is performed by specific histone chaperones. Okay, and same goes for the H2B. Okay, for the deposition of this H2B canonical histones, it requires FACT and NAP1. Okay, most of the times FACT, NAP1, CAF1 and ASP1 is involved in the depositing. Okay, and also this nucleophosphine and the nucleosome assembly proteins. Okay, and apart from that, sometimes this ubinuclein or SR cap or what we call this as HERA chaperone, okay, and also the ERV endogenous ERV chaperones, okay, or some kind of chaperones which are specific for the Z variants, okay, that is CHZ chaperones. Okay, these kind of chaperones, they are rarely seen, but mostly ASF1, CAF1 and FACT will be seen, will be playing in the major role. Okay, so these chaperones, FACT1 and NAP1 chaperones, they deposit the histone to be the canonical histones. Okay, and the deposition of these variants, okay, the histone to be variants is not known. Okay, but the specificity is known. Okay, the histone to be W and TH2. T refers for the testis. Okay, TH2B testis H2B. Okay, so this is specific for the testis only. Okay, this is one. And H4 histone mainly deposited by the CAF1. Okay, and the variant form of this H4 histone is H4G, and it is deposited by the NPM1 histone chaperone. Okay, so NPM1 is also called as the nucleophosphine 1. Okay, and mostly this histone 4 and its variant form G is deposited on the rDNA transcription regions. Okay, wherever the ribosomal DNA transcription happens, okay, at that regions, in that regions, the nucleosome will have this kind of variant forms of the histone 4 proteins. Okay, so this is what about the histone chaperones, and the major role which they do in the nuclear genome. Okay, depositing things. Okay, and if we even talk about the functions of the histone variants, first major chaperone what we have is the H2AZ2, which is involved in the transcription regulations. Okay, and we also have this one H2AX, which is mainly specific for the X chromosomes. Okay, and this is involved in the DNA double stranded break repeats. Okay, wherever the double stranded break repeats occurs, the H2AX replaces the H2A variant, okay, from that nucleosomes. Okay, and uncoils or loosens this nucleosome structure. Okay, that means the DNA will be in the less condensed form whenever the H2A gets replaced by the H2AX histone variant. Okay, and this helps in the DNA double stranded repair break repair by recruiting a lot of polymerases and the ligase enzymes or the what we call it as repair system proteins. Okay, the proteins which are involved in the repair systems. Okay, like methyl transferases or some kind of extra what we call that as the exonucleases and the endonucleases. Okay, so those kind of proteins mainly get recruited by this by replacement of this H2A by the H2AX. Okay, and then we also have this macro H2A. Since this macro H2A is a lot more bigger size, it compacts the nucleosome further level. Okay, and that is involved in the transcription suppressions. Okay, so whenever the actual chromosome consider this is the the actual nucleosome model. Okay, this one. Okay, so whenever this compacts further into very thin form, okay, that means the very highly condensed form. Okay, so no DNA sequence will be available to any kind of polymerases or any kind of transcription factors. Okay, so at that time what happens is the entire gene suppression will be taking place. Okay, so that suppression is mainly due to the macro H2A. Okay, it contains an extra 30 kilodalton domain, that means around 200 amino acid or 28 220 amino acid chain, extra polypeptide chain. So that that kind of the presence of extra 30 kilodalton domain at the C terminal end will make this macro H2A much more bigger size and involves in the heterochromatinization. Okay, and we also have a specific histone 3 variants. Okay, it's 3.1, 3.2 and 3.3. Okay, so these 3.1, 3.2 are canonical histone variant, histone proteins. Okay, that means they only differ in one amino acid. Okay, if we talk about the structure of this histone 3, okay, and the structure of this histone 3.1 and 3.2, these two variants they only differ by one amino acid. Okay, so that means they exactly resemble the structure of this histone 3 variant. Okay, but the amino acid which is different in these two variants will impart the different kind of functions to this histone 3.1 and 3.3. Okay, what I have said you in this point is mammalian H3.1 and 3.2 differ by only single amino acid. Okay, that is serine on the H3.1, and alanine will be seen on this H3.1, while serine is seen on this H3.2. Okay, so this kind of difference will impart the different functional properties of two isoforms. Okay, and 3.3 this histone variant is actually cell cycle independent. Okay, so that means it is expressed throughout the cell cycle. Okay, but this histone 3.1 variant and 3.2 variant they are replication dependent. Okay, whenever the replication happens at that times only their synthesis will be seen. Okay, but in the case of this H3.3 variant, their synthesis is replication independent. Okay, throughout the cycle, so throughout the cell cycle we can see the synthesis of the H3.3 variant. Okay, and we also have this major histone variant that will be seen in the case of the centromere regions. Okay, that is called as centromere protein A, which is also a H3 variant. Okay, and this is an epigenetic marker for the centromere. Okay, wherever the centromere is present we can see this kind of variant. Okay, and it promotes the flexible entry and exit of the DNA regions in the nucleosomes. Okay, so this is what about the major histone variants and their functions. Okay, and the replacement of the H3.3 is found in active chromatin, and H3.3 mainly functions in the germline. Okay, and H2A, as I said it has highest number of known histone variants, while H4 has low number of this known variants. Okay, and in studies in the East, okay, so they have implicated that H2AZ in establishing transcription competence and counteracting heterochromatic silencing and replacing the H2A with H2AZ by ATP dependent nucleosome remodeler. Okay, so there is a remodeler, chromatin remodeler, which requires an ATP molecule. It replaces the H2A with the H2AZ. Okay, whenever it replaces the H2A with the H2AZ. Okay, so that makes the heterochromatic silencing. Okay, that makes the chromatin the chromatin in inactive form. Okay, the heterochromatin form. Okay, highly condensed form. Okay, so H2AZ variants we have H2AZ1 and Z2. They also differ by only 3 amino acids. Okay, 3.1 and 3.2 differ by single amino acids, while Z1 and Z2 differ by 3 amino acids. Okay, and these differences will also impart the different types of functionality and also the distributions. Okay, and in the case of mammalian X chromosomes, okay, whatever the X variants are there, histone X variants are there. Okay, they all be presenting in this mammalian X chromosomes. Okay, so there 3 different H2A variants, phospho H2AX and macro H2A and H2AB. Okay, so they are found in the mammalian X chromosomes. Okay, and they promote the silencing or activation of some kind of genes in the germline. Okay, understood. So they also involved in the dosage compensation. That means representing the half part of the chromosomes in some organisms, or activating the X chromosomes to double level in some organisms. Okay, this kind of doubling the activities of the X chromosomes, or the suppressing the activities of the X chromosome is what we call as a dosage compensation. Okay, and dosage compensation will be seen in differently in different organisms. Okay, and mostly dosage compensation is promoted by these variants. Okay, phospho H2AX, macro H2A and H2AB variants. Okay, and how do we study this kind of the functions of this histone variants or the canonical histone? Okay, so the functioning and positioning of the histone variants can be studied by chromatin immunoprecipitation, what we call as a chip technique. Okay, so in this chip technique, what happens is the nucleosome containing a particular histone variants, they are precipitated by an antibody, okay, which is specific to the specific histone variants. Okay, and can be studied, and we can study the the functioning of this histone variant, okay, which is bound to the antibody, and also the position where it is located. Okay, that is what about the major functions of the histone variants. Okay, and how do the histone variants influence the chromatin stability of the properties? One major thing is about the histone 2AB. Okay, so it contains a truncated or a less length of this acidic patch. Okay, the normal histone 2A. Okay, so the normal H2A family members, they contain a long acidic patch as well as normal version of the C terminal. Okay, but this H2AB, okay, it contains a truncated C termini and also lack an acidic patch. Okay, so due to these properties, okay, so this H2AB will now accommodate a short stretch DNA sequences that is around 100 and 118 to 130 base pairs. Okay, instead of normal DNA sequence that is around 147 base pairs. Okay, instead of this normal DNA sequence, okay, it accommodates very less DNA sequence. Okay, very less amount of the DNA. Okay, so due to this it inhibits the chromatin folding, the actual chromatin folding. Okay, and the deposition of this H2AB promote the active transcription. Okay, conversion of the heterochromatin into euchromatin. Okay, and in contrast, H2AZ, okay, which is also a H2A protein variant. Okay, it contains an extended acidic patch. Okay, that means it contains it promotes more nucleosomal stability. That means more heterochromatinization. Okay, H2AB is involved in euchromatinization because it lacks acidic patch and also inhibits the chromatin folding. H2AZ contains a extended acidic patch and promotes the heterochromatinization. Okay, and the homotypic binding, okay, like H2AZ and H2AZ molecule, okay, 2 H2AZ, like 2 H2AZ, 1 H2AZ is combining with the another H2AZ. Okay, this kind of formation of dimers or tetramers is known as homotypic formations. Okay, so this kind of homotypic molecules or homotypic proteins, they contribute to the heterochromatinization or the heterochromatic formations. Okay, because they enhance the stability of this nucleosomes. Okay, and then heterotypic histones, okay, histone variants, okay, containing one H2AZ molecule and other one which is another histone variant, or the normal canonical histone, that is H2A molecules. Okay, so they are reported to have reduced stability because of the steric hindrance. Okay, so whenever the homotypic forms are present, they have promoted the heterochromatinization. When the heterotypic forms are present, in the case of this H2AZ and H2A, so this kind of two different variants which are presenting in the nucleosomes, okay, as dimers or tetramers. This kind of things is called as a heterotypic. Okay, heterotypic, they promote the, so they reduce the nucleosomal stability. Okay, and the reduced stability is due to the steric hindrance on this polypeptide chains. Okay, whatever the amino acids they contain, the amino acids undergo some kind of steric hindrance. Okay, due to the steric hindrance, the chromatin will be unstable. Okay, and inhibit the chromatin folding. Okay, so this kind of heterotypic will promote the euchromatin again. Okay, euchromatinization. Okay, and then we also have the macro H2A. It contains an extra domain. I've said you, it is much more bigger than the canonical H2A protein. Okay, and it also increases the nucleosomal stability. That means chromatinization, heterochromatinization. Okay, this also promotes the heterochromatinization. Okay, and then we have the CNP-A protein, which is mainly present in this centromere regions. Okay, so it also contains two extra amino acids. Okay, that two extra amino acids, arginine at 80th position and also the glycine at 81th positions. Okay, in the loop 1. Okay, in the alpha helix 1 regions. Okay, and this the presence of these two different two extra amino acids in the loop 1 regions. Okay, it interacts with the other kind of histone proteins which are the neighbor histone proteins. Okay, so these kind of interactions also stabilize the nucleosomal stability and also increase the heterochromatinizations. Okay, so these are about some important points of histone variants. Okay, which and their effect, their effect on the chromatin properties. Okay, and we also have the incorporation of this H3.3 into heterotypic form, that is one CNPA variant is combined with the H3.3 in nucleosomes. Okay, during the replications. Okay, so it functions as a placeholder for later replacement by CNPA in G1. Okay, and recruitment of RNA polymerase 2 and RNA polymerase 3. Okay, placeholder in the sense getting replaced. Okay, getting replaced by something. Okay, so that is called as a placeholder term. Okay, so here what happens is during the G1 phase, this H3.3 will get replaced by the another CNPA protein. Okay, so that means heterotypic form will be converted into homotypic. Okay, So this CNP and H3 point will be majorly seen in this S phase or the synthesis phase, okay, during the cell cycle, okay. And CNPA, which has got replaced by H3.3, okay, now forms a homotypic dimer with another CNPA, okay. So this kind of a homotypic dimer will be seen in the G1 phase, okay. So in the G1 phase we will be seeing the homotypic forms, and in the S phase we will be seeing the heterotypic forms, okay. These kind of presence of two different form in two different phases also imparts the nucleosome stability, okay. So in one phase nucleosome will be very stable, and in other phase the nucleosome will be slight stable, okay. And these histone variants because they contain different type of amino acids and extra acidic patches or truncated versions of the acidic patches, they impart the nucleosomal stability or the chromatin stability, okay. So this is what about the stability of the the histone variants impart how the histone variants impart the stability to this the chromatin molecule, okay. So this the presence of the H2AB or the H3.3 with H2AZ, okay. So this is what we seen in the previous slide, okay. So this H2AB it promotes the euchromatinization, that means decrease in nucleosome stability, okay. And because it contains less acidic patch, okay? And H3.3 and H2AZ they also contribute to the euchromatinizations because they also destabilize the nucleosomes, okay. And this kind of presence of multiple variants, which we often call as a heterotypic dimers, okay. And increase in nucleosome stability, mainly seen in the case of H2AZ variant, okay. If it is present, the nucleosome will be in the more stable form. And macro H2A, because it contains an extra domain, this one, okay, the extra amino acid chain length, okay. So it promotes the stability of this nucleosomes, okay. And we also have this centromere protein, okay, and H3.3 and centromere heterotypic protein, okay. So this dimer, histone dimers, they also promote the nucleosome stability, okay. And I've said you this H3.3 functions as the placeholder. It involves in the placeholder functions, okay. H3.3 in the G1 phase it gets replaced by only another CNP proteins, okay. When it gets replaced by another CNP proteins, okay, the stability will get decreased now and converts into euchromatinizations, okay. So this is what this is how the histone variants impart the stability of this chromatin or nucleosomes, okay. And that is how the stability is stability gets affected, okay. And then we have the linker histone variants. Nowhere we have talked about the linker histone variants, okay, because lot of the linker histone variant H1 variants they contain different domains, okay, that we call as a winged helix domain, okay. Because of this winged helix domain most of the researchers haven't figured out any kind of functions on this functions for this H1 variant, as well as the variant functions of the variant forms, okay. And mostly these H1 variants they are dynamic, okay. Most of the H1 variants are dynamic. That means they change from period to period, or the genes from particular gene sequence to particular gene sequence, okay. We won't be seeing H1 variants throughout the cell cycle, okay. At particular phase only we will be seeing the H1 variants, okay. So in order to extract the histone variants, or in order to know about the functions of the H1 variants or histones, canonical histones, H1 variant, linker histones, okay, that is a kind of difficult job, okay. Even though humans and mouse cells, they are known to contain histone variants, okay. We have 11 H1 variants, okay. What we have discovered is 11 H1 variants, okay, and they are encoded by single genes, okay. Only one gene will be involved in synthesizing this 11 histone variants, okay. And 11 histone variants are due to alternative splicing of a single mRNA transcript, okay. And 6 variants are mainly expressed in the S phase, okay, while other 5 they are expressed throughout the cell cycle, okay, whole cell cycle, okay. And the location, if we talk about the location of these genes which are involved in production of the H1 variants, they are located as cluster on human cell chromosome 6, okay. Chromosome 6 contains the histone cluster, linker histone cluster 1, okay, and it encodes for 11 variants, okay. So 6 are expressed on the S phase, while 5 are expressed throughout the cell cycle, okay. So most of the functions are not yet clear, but we know, some functions of the H1 variants, okay, like H1 variant, especially the H1.2, okay. So it is involved in the DNA damage and repair, okay. So whenever there is a damage on the DNA sequences, H1 is replaced by the H1.2 and modulate the repair of the DNA damage, okay. And we also have this H1.0, which is mainly found in these differentiated tissues, as well as this the sperms and the majorly in the sperms, okay. The normal linker histones are replaced by some kind of histone variants, which we often call as the protamines, okay. The protamines are kind of very basic amino acid rich proteins, okay. More than uh, histones, they are quite highly basic, okay. Whatever the basic amino acids are present in the normal histones, okay, protamines contains twice of that basic amino acids, okay. And overexpression of this H10 and H1C, which are also linker histone variants, okay, results in decreased rate of chromatin digestion by micrococal nucleus, okay. So micrococal nucleus, they breaks the chromatin threads into simple nucleosome threads, okay, that about 10 to 20 nucleosome threads, okay. So this kind of chromatin digestion by new micrococal nucleus will be inhibited by this H10 and H1C when they are overexpressed, okay. So this is about the linker one histones, okay. And I've said you the N-terminal tails of these histones, when they are forming the nucleosome cores, okay, so they get extruded out, okay. That means from one nucleosome the N-terminal tails will come out and links and get linked with neighboring nucleosome core, okay. If you consider this is a typical nucleosome core, okay. So from this these are what the nucleosomes are. So from this nucleosome from this histone core protein, from this histones, okay, the core histone proteins, okay, or the core octamer, then tails will extrude out like this, the internal tails, okay, and they get linked with neighboring nucleosomes, okay. This kind of linking actually uh, provides an extra stability to the nucleosomes, as well as to undergo a lot of covalent modifications, okay. And N-terminus tails, N-terminus, okay, the internal regions of this histone proteins, okay, it is responsible for the gene activity, okay. And this N-terminus mainly contains 7 to 100 amino acid residues, okay, typically, okay. And sorry, this 30 amino acids, okay, 30 amino acids will be present in this N-terminus tails, okay, and they are involved in the gene regulation activities, so for the maintenance of this heterochromatin and euchromatin states, okay. And in the major modifications which are seen in the N-terminus are the methylations, acetylations, and phosphorylations and all these things, okay. And mostly apart from the modifications, okay, proteins like HP1 which is involved in maintenance of the heterochromatin state, okay, and the polychrome group proteins, okay, and also the chromatin remodeling complex will be recruited on this N-terminus regions, okay, and perform the modeling activities, so called the modification activities, okay. So these kind of things will be seen in this N-terminus, okay. And they also regulate the DNA accessibility, okay, DNA accessibility to the polymerase enzymes, okay. So if the polymerase want to bind to the DNA sequences, first the histones has to get replaced, okay, or some kind of modification has to done on the histone N-terminus, so that the stabilization of the chromatin molecules will get reduced, and the polymerase the RNA polymerase or the DNA polymerase will get bound to this DNA sequences, okay. And post translation modifications I've said you which will be seeing on this N-terminal tails, okay. So histone variants also undergoes the post translational modifications apart from the canonical histones, okay. And most of the post translational modifications will be done by this histone chaperones only, okay. So one of the major post translational modification is done by this ATRX protein, that is arabidopsis triethioacrylated protein, okay. So it specifically methylates H3.1, which contains an alanine amino acid 31st position, okay, and not H3.3, which contains a serine amino acid is 31st positions, okay. So these two amino acids, which are histone 3 variants, they differ only by a single amino acid, that is alanine and serine, okay. Due to this difference, this ATRX protein only works on the 3.1 variant and methylates the alanine amino acid. Okay, so this kind of methylation is very specific by the histone chaperones. Okay, and we also have another type of post-translational modification. Okay, that is a phosphorylation. Okay, or the second type of modification is variant specific. Okay, so which is also called as a variant specific modification. Okay, so this type of variant specific modification, so if it is happening on H3.3, specifically on the serine amino acid. Okay, so when serine amino acid undergoes phosphorylation, okay, it increases the acetyl transferase activity of the P300. Okay, so this P300 protein, okay, so it transfers the acetyl groups onto the H3K27. Okay, so the lysine amino acid. So this lysine amino acid undergoes acetyl transferase activities. Okay, when this P300 acts on it. Okay, so this P300 activity is will get increased when this H3.3 gets phosphorylated. Okay, so the serine amino acid which is present on the H3.3 histone variant, when it gets phosphorylated, it promotes the activity of this P300 protein. Okay, so when the activity of this P300 gets promoted, it phosphorylates it transfers an acetyl group onto the lysine amino acid. Okay, lysine amino acid which is placed on 27th position on this H3 histone. Okay, so that get acetylated. Okay, so this kind of acetylation, uh, indirect acetylation by this P300 protein. Okay, or by this the variant specific protein modifications, or the chromatin associated enzyme activity. Okay, so this is another specific post-translational modification, or the another mechanism of this post-translational modification mechanisms. Okay, and this kind of modification will mark the transcriptionally active chromatin. Okay, so convert the euchromatin into active chromatin. Okay, and in turn, the lysine amino acid which is placed on the 36th position of this H3.3 undergoes trimethylation. Okay, that means 3 methyl groups will be added on this variant. Okay, so that is mainly done by this zinc finger MYD type containing protein. Okay, so this is actually reported as a repressor protein. Okay, so this is the repressor for transcription elongation. Okay, whenever the RNA polymerases perform the transcription process. Okay, so that during that time the elongation process will be inhibited by this protein. Okay, by doing the trimethylations on the lysine 36 the lysine amino acid. Okay, and because of this the activity, the phosphorylation of the serine also happens. Okay, the H3.3 linker histone also contains the serine amino acid on the 31 positions. Okay, when it undergoes phosphorylation by this ZMYND11 protein. Okay, it regulates the transcription. Okay, the transcription process. Okay? The transcription regulation is likely multifaceted. Okay, that means very complex. Okay, very complex transcription regulation will be seen. Then we have this H3.3, the incorporation leads to death the domain associated protein. Okay. And another mechanism is recruitment of different types of proteins indirectly, as in the case of this crab protein. Okay, which has got recruited which has got recruited dependently by this death domain associated protein, which you also call it the DAX protein. Okay, so this protein will be seen in the apoptosis process. Okay, so this H3.3. Okay, so it actually gets associated with this DAX protein. Okay, and this DAX protein will link this Krupel associated protein, or the crab protein, which is further associated with the set DB1. Okay, which is also called as the set domain bifurcated 1 protein. Okay, so presence of all these protein, okay, makes the chromatin to be less stabilized, or what we call it as to make the chromatin to loosen the DNA sequences to make the gene transcription to happen. Okay, so this is what happens, the incorporations of the direct dependent proteins or the indirect proteins. Okay, and in another mechanism which we also call as a combinatorial readout. Okay, what happens here is presence of an acetyl group on one histone protein. Okay, like on the H4 protein. Okay, H4 is acetylation, okay, acetylation on the H4 protein, okay. So it couples with another histone variant, that is H2AZ1, okay, and it promotes the recruitment of another bromodomain containing protein, which further promotes the activation, transcription activation, okay, the promote the repression of a gene, okay. So this is what happens in the combinatorial readout, okay. Presence of one acetyl group on one histone protein will combine with a, or will couple up with another histone variant, okay. So this kind of coupling will recruit some kind of a bromodomain containing protein or any other kind of histone chaperone, okay, and regulate the transcription activation, okay. So this is what happens, okay. And there is also site specific post translational modification will also be seen, okay. So that means, whenever this H2AZ protein is formed, so when it is present in the euchromatin, okay, so it stays in acetylated form, okay. So the acetylation on the H2AZ will be done in euchromatin, okay, and the same H2AZ undergoes methylations and the ubiquitinations when it is present in the heterochromatin, okay. So in the heterochromatin, the H2AZ is ubiquitinated as well as methylated or methylated, and in the case of euchromatin, this H2AZ is seen as methylated, acetylated form, okay. So acetylation will promote the euchromatin, and methylation and ubiquitination promote the heterochromatin, okay, and it also contributes the gene activity and the silencing, okay. So these are about the histone variants, okay, normal general histone variants and their post-translational modifications, okay, specific modifications, okay, and the mechanisms or different kinds of mechanisms of the post-translational modifications, okay. And we also have the canonical histones like H3 and also the H2A and H2B, okay. These are the major canonical histones which undergoes modifications, okay, and most of the modifications are seen on this H3 histones only, that is methylations, okay. Methylations are also specific, okay. If methylations happen on the lysine 9 or 9th lysine amino acid, okay, on the H3 histone, okay, it promotes the gene silence, okay. That means, it converts the euchromatin to heterochromatin, okay. If the same methylations, if they are present on the some other lysine amino acid, which some other position of the lysine amino, that is, on the 4th position of lysine amino acid, okay, that promote the converting of heterochromatin to euchromatin, okay. So conversion of hetero to euchromatin will be done by methylating a lysine amino acid that is present on the 4th position, okay, on histone 3, okay. The same methylation, if it is, happening on the 9th position, the lysine amino acid, which is a part of histone 3 protein, okay. So that convert the euchromatin to heterochromatin, okay. And most of the methylations will be done by set domain containing protein, that is SUV proteins, or set 7 or 9 proteins, okay. All these are MLL proteins, okay. These are all set domain containing proteins, and we have also seen the role of this LSD protein, okay, which you also call as the methyl transferase, okay. This protein transfer the methyl group, okay. So all these proteins, all these enzymes stays under the methyl groups, okay. Some of them are the set domain containing, and some of them are the DNA methyl transferase family proteins, okay, and some of them are the SUV proteins, okay, the chromodomain containing proteins, okay. So all these things, okay, they transfer the methyl groups on the specific lysine amino acids, okay, specific position to lysine amino acids, okay. So this kind of different amino acid promotes the different conversions, okay, either converting a heterochromatin to euchromatin, or converting a euchromatin to heterochromatin, okay. Both happens, okay. A gene silencing will be seen, and as well as the gene activation will be seen, okay. But the methylation patterns will be different, okay. Transferring of this methylation, differently methylated histone proteins, or the differently methylated DNA from parent to offspring is often called as epigenetics, okay. We are not doing any kind of mutation, we are actually modifying, okay. And so apart from the methylations, we can also see the ubiquitination, phosphorylations, and acetylations happening. Most of the acetylations, they involve gene activations, okay. Wherever they happen, they involve the gene activations, and sometimes acetylations also marks the DNA repair or histone depositions, okay. And methylations, it will be involving both, okay, repression and activation, gene activations, okay? And thus, depending on the lysine amino acids, okay, the position where the lysine is present, okay, so that position will tell you whether the gene has to undergo activation or the repression, okay. And phosphorylations, it is mainly involved in the apoptosis, DNA repair, mitosis, okay. And remember this point, this only one H2B serine amino acid which is present in the H2B at 14th position, okay. If it undergoes phosphorylation, it is mark for apoptosis, okay. That means apoptosis is about to happen inside that cell, okay. So this kind of post-translational modifications on the serine amino acid which is present in the H2B protein, okay. and H3, normal H3, canonical H3, if it undergoes phosphorylations, okay, at serine 10 or 311 and serine 28, okay, so they will be involving in the DNA repair and the mitosis process, okay. And T45, 345 on histone 3, if it undergoes phosphorylation, that marks for DNA replications, okay. And most of the modifications, they either indicates a gene activation or the gene silencing, okay, and also DNA repairs, okay. And another specific, or the unique histone modification, that is ubiquitination of this lysine amino acid at 23rd position on the histone 3. It is involving in maintenance of the DNA methylation patterns, okay. That is in this kind of histone 3 modification majorly seen in the CpG islands, okay, or the CpG sites, okay. So these are about the most common mammalian histone post-translational modification what we see in the nuclear genome, okay. So this is about the today's class.